So let's go ahead and kind of talk through some of these big key concepts on this study guide. For those of you that are at home, I welcome you to pull up the study guide on Google Classroom to kind of follow along if you don't have the paper copy here. So the structure of DNA. We do need to know kind of the basic layout of what this looks like here. So basically on the outsides of your DNA strands, okay, that is going to be made up of sugar and phosphate. So they're kind of alternating. Okay. So real quick, Mike, could you give me a thumbs up if you can see what I see? Okay, perfect. Thank you. Yeah, could you mind coming up and grabbing it? Got some up here if you guys change your mind. Okay, so in the middle of our sugar phosphate backbone, we have our bases. So the bases are going to be in the middle. And remember, those are A's, T's, C's, and G's. Okay, they're held together in the middle with hydrogen bonds. Okay. Now, I am going to move through this pretty quick because I want to try and at least touch each of our big key concepts. So if there's anything that you miss, feel free to stick around after class and I'll have my key up here. So that just says hydrogen bonds. To be honest, number 2 on here, do not need to worry about it. That was a concept that we actually didn't end up having time to get to during our first unit, so I'm just going to nix that part. Okay. So, what's the role of each of these different parts in the cell? And I'm going to be very brief here. So, the role of DNA is to store genetic information. The role of mRNA is to move the copy of DNA, which is mRNA, into the cytoplasm, okay, where it takes it out to the ribosome. Okay. tRNA is going to bring the amino acid to the mRNA and the ribosome. So we'll need to have a pretty clear understanding of the process of transcription and translation. Unfortunately, I probably won't have time to walk through all of that here while we're going through this. So you might need to kind of review that on your own. So you will need to be able to create a DNA to DNA copy, so making a complementary sequence of DNA to DNA. And you will also need to be able to make a copy from DNA to mRNA. So for every, we think back to this here, for every T on DNA, we're going to have an A. For every A on DNA, we're going to have a U. For every G on DNA, you're going to have a C on mRNA. So if we continue this, we're going to have G, C, U, G, A, G. And I see that some of you guys are messaging me on Remind, which is fine, but just know I'm not going to be able to respond until we're done going through this here. Okay. So we need to use these codon wheels to figure out what amino acids we're going to have here. Okay. So for this one here, we have AUC. We would go A, U, C. That's going to be isoleucine. Remember, we can just abbreviate that with the first 3 letters. Okay. Next one, GCU. GCU. That's going to be alanine. ALA. GAG. GAG is going to be glutamic acid. So we need to be pretty comfortable with how to do that there. Okay, so like I said, we're going to move pretty quick here. All right, DNA versus RNA. We need to know the differences between those. DNA is inside of the nucleus. RNA technically can be in the nucleus, and it's also going to be in the cytoplasm. How many strands does DNA have? 2. What about RNA? Yep. DNA is going to have A, T, C, and G. RNA is going to have A, U, C, and G. Okay. So, for transcription versus translation. Okay. Transcription takes place in the nucleus. All that we need for transcription, for our intents and purposes, is DNA. Technically, we also need RNA polymerase, but to be honest, we didn't really talk about that too much. Okay. And then the output, what comes out of transcription, is going to be mRNA. So translation, that's going to be happening out in the cytoplasm, which you could also say the ribosome, that would be fine too. And for translation to take place, we need mRNA, we need tRNA, we need the ribosome, and sorry we're getting a little tight here, we're also going to need amino acids. And what do we make at the end of it? The process is protein synthesis. Yep, we're going to make proteins. We actually got a lot done in 5 minutes. Maybe I might slow it down and go a little bit more in depth here, especially for these next two. All right, so thinking about the process of transcription and translation. Okay, transcription, first step is that DNA unzips. Second step, which we didn't really talk about this part, so I'm not expecting you guys to know what RNA polymerase is. That's fine. But RNA polymerase makes a copy of DNA onto mRNA. And then the mRNA leaves the nucleus. What's up? Are you talking about on the test? Yeah. No. K's either. Because frankly, a lot of the stuff is straight out of our notes. So I wouldn't. Yeah, it's all multiple choice. Does that make sense? Okay, so now for translation, okay, the mRNA finds the ribosome, then the matching tRNA, sorry, this is a little tight here, I should have put another space there. So the matching tRNA brings the amino acid to mRNA. And I'm just going to streamline this to saying that the growing amino acid chain builds a new protein. All right, so what is the molecule that is within a dose of the COVID-19 vaccine, which that should say Pfizer or Moderna, and that is going to be mRNA for, oops, that's actually kind of the next answer, sorry. So we can just say mRNA. That's technically let's actually add viral mRNA. That's more technical. All right, what is the protein that is made from your body after you receive the COVID-19 vaccine? So that is a viral spike protein. And then what cell process makes this protein? That's going to be translation. All right. Okay. Describe how your immune system responds once the protein is made in your body from the previous question. Okay, so the viral spike protein, let's actually call this number 1. So viral spike protein is taken in by memory B cell and let's say, I'm going to zoom this out a little bit, and a matching antibody is made. Okay. Antibodies go out and find COVID-19 virus and surround it. Surrounded virus is destroyed by white blood cell. Let's see if that fits. Yeah, perfect. I know, we're cruising, right?
We're definitely cruising. We're doing the opposite.
We're bruising. All right, so going back to bacteria and viruses here. So both bacteria and viruses are going to have genetic information, but bacteria are going to be living, viruses are non-living. Bacteria are going to be treated by antibiotics. Viruses are going to be treated with vaccines. All right, so our 3 lines of defense, your outermost defense is going to be your skin, your physical barriers. Your second line of defense is going to be fever and inflammation. And then your third line of defense is going to be your antibodies and immunity. All right, so these next couple ones here. Okay, now we're moving into our current unit. So, is your mask red? I don't know, but we gotta get going. Are you doing a bloody nose or something? Okay, yeah, go for it, bud. You're fine. You're good. No worries, man. All right, so if we're looking at these two graphs, the difference between a polygenic and then a single gene trait. This one is going to be a single gene trait. This one is going to be a polygenic trait. Simply because there's more than two different possible traits that you can have. This one only has two. Technically, this one's talking about single. This one's talking about polygenic. All right. Okay, different types of mutations here. All right. So I'm going to make these real quick. So a substitution is going to be a switch. Insertion, okay, you can see insert there, means that something was inserted in between. Deletion, you could probably guess that. That means that one was deleted. Frame shift means everything moves over one. And a silent mutation means nothing changes. Insertion, deletion are going to lead into frame shift mutations. Okay. So if we look there, we kind of already went through how to convert from DNA to mRNA to amino acids. So I don't think we're going to focus on that too much. But what we look when we see this, is we need to identify where the change was. So if we look, this G is no longer present. So this is what we call a deletion. All right, and please don't pack up, guys. We're going to go down to the wire here. Okay, so looking at these ones. Okay, oops, sorry, I'm going to move this down. Okay, so looking at these ones here, we've got long whiskers being dominant over short whiskers. So if we have a homozygous dominant seal, big W, with a heterozygous seal, okay, that means that we are going to have 50% big W, and then 50% big W, little w. Okay. So phenotypes would be 100% long whiskers. So within a population of butterflies, we've got big B brown dominant over little b white. So if 40% are white, that means 40% are little b. 38% are heterozygous. Okay. So what does that mean for the percent that is big B here? How can we figure that out? So we add them up, yes, but then what do we do with that number? Right. So if we add those up, that's 78. So yep, it's going to be 22% big B. So 22% big B. So big B little b is half big B, half little b. So we're just going to divide that in half. So that means that it's 19% big B and 19% little b. So for the frequency of the big allele, 22% plus 19%, okay, that's going to give us 41%. Same thing for little b, 40% plus 19, it's going to give us 59%. So last but not least, if the brown allele, so big B, changed to 44% and it was at 41%, did evolution take place? Did it change?
Yeah.
So yes, evolution did take place. All right, so in this case, yes, evolution did take place. Because the allele frequency changed over time. Okay. So if we have a population of 70 monsters and long hair is dominant over short hair, 36 of the heterozygous long hair, 36 of the monsters are heterozygous for long hair, and then 20 are going to have short hair. So our total is going to be 70 monsters. 36 of them are big L, little l. They're heterozygous. So when we plug this in, we're simply just going to do 36 divided by 70, and that's going to give us 0.51. Okay, and if we think about that there, if we want to convert that to a percentage, which you could leave it as a decimal and just make sure that it adds up to 1.0, that would be fine too. But if we want to convert this to a percentage, we're just going to multiply it times 100. So that gives us 51% big L, little l there. Okay. All right, so now it's also saying that 20 of the monsters are going to have short hair. So they're going to be recessive, little l, little l. So we'll do the same thing, 20 divided by 70. And that's going to give us 0.285, which we could round that to 0.29. Again, if we want to turn that into a percentage, multiply it times 100. That actually gives us 29%. Little l, little l. So we need to still figure out what percentage is going to be big L, big L. So 51% is heterozygous, 29% is homozygous recessive. We're just going to add those two up. So that gives us 80% between those two. If we subtract 80 from 100, that's going to leave us 20%. Okay, so the same thing we did before, we would just go and divide the heterozygous by 2, so we're going to divide that by 2. Okay, so that's going to give us 20.5, which I know that gives us a 0.5%, but that's okay. Oops, sorry, it would be 25.5. And that's going to be 25.5 for big L, 25.5 for little l. And then we're just going to add those to each of these here. So plus 25.5% here, plus 25.5% here. Okay. And that's how we're going to go ahead and solve for that. You'll just go ahead and add those together. Okay. Now, I'm not going to go into all of the different parts of how we would write a CER, and we're not going to go too in depth into the different processes of evolution because, frankly, we just talked about them together in class. So for natural selection here, natural selection is when traits are selected for or against in an environment. So this could be from disease, this could be from a predator seeking out a certain color of prey, for example. This could be from certain types of behaviors that allow some organisms to be able to attract a mate. Okay, so descent with modification means that offspring both have traits of their parents but are also different. So, for example, if you have two parents, they're going to combine their traits in their offspring. So their offspring will have some of the traits of each parent, but they won't be the exact same as their parent. And then finally, common descent is the idea or the theory that all organisms on Earth have descended from a common ancestor. Nope, I'm going to run out of space. Common ancestor. So the theory that all organisms on Earth have descended from a common ancestor. Okay. So I'm not going to go through, like I said, and write a formal example of this, but I can kind of highlight what the key things that we would be looking for this. So write one piece of evidence that supports the claim that white fur provides an advantage in Arctic conditions when wolves are present. So what we could do is we could just pick out a couple. Remember, if we're writing an evidence statement, we need data. So we could pick out and say that in the Arctic, okay, we had white fur rabbits at 5 in the first generation, and then 550 in the last generation. Brown fur was 1 in the first generation in the Arctic environment. And then by generation 7, they had gone extinct in that area. They were all killed off. And then in the desert environment, we could say that there were 2 white fur rabbits in generation 1. And then there was only 10 in generation 7. And then for brown fur, we could say that there were none in generation 1, and then there were 80 in generation 7. So when we're providing a reasoning, we need to basically say the data. We need to show the data. So in the Arctic, let's say this environment, the white fur increased over the 7 generations. And we could also say, and the brown fur decreased. So this is the trend. Oops, sorry. This is outlining the trend there. So now we could say this happened because the white fur camouflaged the rabbits. Couldn't remember if they were rabbits or bunnies. Let's just say rabbits. Yep, they were rabbits. So the white fur camouflaged the rabbits in their surroundings, in their Arctic surroundings, let's say that. And the wolves were unable to see them as easily. Okay. All right. So again, remember, for data or evidence statements, we can only include numbers. We don't include any of this explanation in there. Okay. All right. Oops, let's zoom that up a little bit. Okay. Last but not least, we're looking at antibiotic resistance and then examples and evidence for evolution. Okay. So let's go ahead and take a look at this here. So if we look here, we've got these different populations of bacteria that exist inside of, let's say that this is inside of someone's body. And these were all exposed to the antibiotic tetracycline. Okay. So if we look here, as we notice, okay, the B. farfalle, the E. rotini that's plain, the E. rotini that's orange, and then the E. rotini that's green, all of them changed over time. So if we look here, going from beginning to end, the farfalle decreased, the rotini slightly decreased, sorry, the E. rotini that's plain slightly decreased, the orange rotini slightly increased, and then the green rotini dramatically increased from beginning to end. Okay, so most likely the E. rotini is going to be the most resistant to the antibiotic, even though these other ones showed some resistance because they were able to survive. But the B. farfalle was killed in very high amounts. So the E. rotini is most likely the most resistant because it has the highest survival rate. Okay. So antibiotics are an evolutionary process because natural selection is essentially taking place here. So natural selection is, let's say, is applying pressure, it's applying selective pressure on the other species besides the green rotini. And this is going to cause a change over time. And anytime there's a change over time, that means that evolution has taken place. Looking at the pieces of evidence for the theory of evolution here. Okay. So I'm actually going to start with this. So the key idea that the theory of evolution supports is going to be the idea of common descent. Okay. So common descent is supported by the theory of evolution. So we see all of these different pieces of evidence that support the fact that all our organisms on Earth descended from the same common ancestor. Okay. So we've got a couple pieces of evidence that we can use here. and I'm going to be very brief in my descriptions. So we could look at things like the fossil record. So that's going to be looking at what we call transitional species. If you remember, we were talking about the land mammals that eventually turned into whales, right? So there were a lot of organisms within our evolutionary history that had bones and things that basically showed us kind of in between species for between the land mammals and the whales. So we see these all over the place. We've got Archaeopteryx, the bridge between dinosaurs and birds. So there's a lot of transitional species in the fossil record. That implies this change over time. So we also have embryological similarities. This means that when we are developing as embryos, all vertebrate embryos are going to look the exact same when they are developing. Okay. So then we also have homologous structures. So these are going to be, for example, like the bones of animals in the arms. So the bones that are in the forearm of a human and a bat and a whale, the bones are going to be the exact same structure, but they are going to have a very different function. Okay. So we also have things like vestigial structures. So these are leftovers like a whale hip bone or a human appendix or a human tailbone. So they no longer serve a function. Okay. And then last but not least, we have biochemical evidence, basically looking at the DNA similarities. So humans and chimpanzees, for example, have very similar DNA. So we can use those to compare here. Okay. So our last topic is going to be our examples of evolution. If we remember, we talked about the 5 fingers of evolution. Okay, so let's go from one end of our hand to the other. So right thumb meant thumbs up or thumbs down for natural selection. Okay. Your pointer finger, that was going to be gene flow. So we have things moving around, which, wow, my finger looks really weird lagging like that. So that's going to be gene flow. Basically, that's your pointer finger, and that represents organisms moving in and out of different environments. Okay. Your middle finger, that was for mutation. Okay, so middle finger stands for mutation. Those are new traits that arise in a population. Your ring finger is for speciation. Or sorry, not speciation. Selective mating. Getting my terms confused here. Selective mating. Okay, and that means that organisms are choosy, that they can choose a mate based on certain behaviors. So, for example, the peacock. A female is much more likely to pick a male peacock that has a larger plume of feathers. And then finally, your pinky finger is going to be for genetic drift. Now, for genetic drift, we're talking about, sorry, I probably should have added that there. We're talking about small populations. And the two examples of that were bottleneck, which means that there's a large die-off of organisms. And then founder's effect. Basically, there's going to be a small colony that starts in a new place. Okay. And to be honest, for this one, I kind of explained the keywords to identify each example all together up here. Okay. So I apologize that we didn't quite have enough time to finish all this, and probably this is better because I was able to go through in a little bit more detail anyway. But yeah, so you have all this material at your disposal to help you prepare for the final exam. So now you have your key to the study guide. You also have the review on Google Classroom. So if you have any questions, please don't hesitate to reach out. And thank you guys, and have a fantastic day. We'll talk to you guys soon.